So this is what we've already been going through. Kind of a reminder. An allele is a version of a gene. Sometimes there's only one version of a gene, which means that it's gone to fixation, and any other version is has less fitness. So an allele is either from mom or dad. They sometimes have the same allele, oftentimes different. And of course, those alleles on those bands that we were looking at, those are alleles. So, which patterns can't belong to a child of this couple? So we've got dad here. Got dad here, there, and there. Got mom up here. And let's see here. Oh, I see. So which one is it? C. Okay. Okay. Okay, has to be D. So this allele right here we have what's called a double band. That means we have the same allele. Same thing up here, we've got a double band. So mom and dad share this same allele. Here, mom doesn't have this, but dad does, but it's a double band. So if you had a double band there, it means the child got both alleles from the father. You can't have that. So it's D. So what kind of cell is this? Eukaryotic. Okay, good. Now remember, all eukaryotes have a nucleus, and they often all have mitochondria and all this other stuff. Now remember we talked about a prokaryote, they don't really exist. They're either bacteria or archaea, neither of which have a nucleus. So if we were talking about a bacteria, it would be generally a lot smaller. Okay? If it was a virus, it'd be smaller still. Except for some new viruses that have been found that are gigantic. Who are these guys? Okay, good. 1953. So complementary base pairs. Okay, and you guys got it. Good. So, which enzyme is used for this? DNA polymerase. Okay. So it adds the complementary nucleotides to a single strand here. So that's how you can actually copy a cell when it needs to be copied. So what procedure will they use to amplify a small amount of DNA to a large? Okay, PCR. So, why are STRs so useful? Well, here's the repeats. This person has 3, 4, 7 repeats. Can we differentiate this person from someone else? Someone else might have 8, or 9, or 3 repeats. And so we can use these STRs to figure out if someone is actually a possible suspect or not. Here's gel electrophoresis again. So we take these different fragments or samples from different people. So one sample per well, put them in, charge it. The shorter fragments go faster, further down. The longer move more slowly. So a chromosome is, of course, one long DNA molecule. Each chromosome, one long DNA molecule with millions of bases or nucleotides. Now, this is a homologous chromosome, of course. So, one from mama, one from papa. Here's the gene. You want to duplicate. And then genes make proteins. That is, a gene is a stretch of DNA that is translated into a protein. Now, a gene really doesn't do anything per se. All it does is it is used as a template for making the protein that you want. Okay, so we've done a little bit of this, but let's take a look. Each of these is an amino acid. A protein is essentially a string of amino acids. Depending on which amino acids are in which place, often determines how the protein is folded and exactly what are the active sites. So what happens is the gene on the chromosome is translated into these amino acids. Now, okay, so here we go. This is the nucleus. Okay, all inside. The DNA is inside the nucleus for a eukaryotic cell. Bacteria, it's not. There is no nucleus. So transcription and translation happen with the bacteria together. Here they're separated. So what I mean by transcription is that this is a cytoplasm. That's the outside of this. That's not the outside of the cell, but it's the outside of the nucleus, where all the mitochondria are. And inside is the DNA. So what happens is the DNA has to be transcribed into a single strand of RNA. So it goes you go from DNA to right there, RNA, and this is mRNA, which means messenger RNA. The messenger RNA is an exact copy of the DNA, but it's a complement of it. Okay? So, at some place you have, let's say, a T, it's going to be, for RNA, it's going to be, we'd say it's normally going to be an A, but actually it's called uracil. It changes a little bit in RNA. But the main thing is you go from DNA to RNA, and this is called okay. Well, let's get this over here. Okay. Now, this messenger RNA, which is a complement to one of the DNA strands, it moves through this pore. So there's ways to get things in and out of a nucleus, and it's a protein pore. And here's the mRNA. And it's now in the cytoplasm. Now, it turns out here are these amino acids, free amino acids, and we have this ribosome. Now, the ribosome is basically an RNA machine, and it's made of RNA. Therefore, what it does is it takes the amino acids using the mRNA as a template and synthesizes the protein. And that's called translation. Okay, and then we go in the ribosome. This is tRNA, and then this goes to protein. And we'll say right here. So we go transcription to translation. Transcription happens inside the nucleus. Translation happens in the cytoplasm. Amino acids are essentially peptides, and they're strung together in a long polypeptide chain. Like this. And this is what the protein actually is. Okay. So this is the basic process by which DNA is turned into a protein. And proteins, remember, do all the work. There are structural proteins. There are proteins like enzymes that handle chemical reactions. There are signaling proteins. Everything that is in our body is all proteins. And those proteins, sometimes they can be long chains, sometimes they can be short, sometimes they can be isomers or of each other, slightly different. And of course, this is where variation comes in, ultimate variation in terms of mutations. Okay. And actually, this should be a RNA in here. And that's transfer RNA, and we'll get to that now. Okay. Now, transcribe. Kind of like a court reporter. So we're taking some code that has information in it and turning it into a way that it can then be used. Translation, and in this case, translation is making amino acids that then become proteins. Let's say from one language to another. I want to do one thing. Okay. Okay. These are nucleotides. It won't work. Okay. We'll say that's an A. Say this is a G. This is a C. T, T, A, G, G, A. Okay. Now, you need 3 nucleotides to make an amino acid. One won't do it. So, here we've got 3 nucleotides. It's called a codon. And this amino acid AGG, or this code, will make an amino acid, whatever it is. Okay? And Remember, these are peptides that then are strung together in proteins. So we've got some amino acid here. Here we have another amino acid. And here we have another amino acid. So we've got 3 amino acids stuck together. Each 3 is a codon. You need 3 to make an amino acid. Now, occasionally, I showed this earlier, where we looked at reading frames. So a reading frame is always going to be a 3 DNA letters long. If it stays a reading frame, that's good. If we were to actually start instead right here, let's say we had a mutation, then we'd end up with a different amino acid or maybe a stop codon. And probably everything downstream of this would all be gobbledygook. So now let's say we do manage to stay in the reading frame, like right here. But let's say we've deleted 3 amino acids. And now we go from here to here. It means that we've still got amino acids that make sense. They're not nonsense proteins, but they're different. And so it's a mutation that might change function, it might not. Depends. Okay. So when Watson and Crick discovered the structure of DNA, they didn't know how to read it. They knew they had the structure of it, but they didn't actually understand about codons, like triplet base pairs, how that translated into RNAs and then into proteins. And but then they found out, in fact, it was Crick who actually found out all those different things. And people thought, well, that's pretty much how things happen. You have different DNA, you get different amino acids, different proteins, different functions. Some proteins work differently than others and might be favored in one environment versus another. But they didn't know anything about what's called gene expression or regulation. That means you're regulating how much DNA is turned into proteins or how much protein you're making. Sometimes you want a little bit of protein, sometimes you want a lot of protein. Let's say an enzyme like lactase, which breaks down milk from a polysaccharide into a monosaccharide, right? Which is a single sugar, glucose. Now, until, I don't know, maybe 10 years ago, we knew virtually nothing about gene expression and regulation. Theoretically, we thought it might be important, but we didn't have the tools to actually look at it. And then slowly we developed. And so we're kind of in a renaissance now, where we're finding out things that we never knew before. Now, one of the things that gene expression does is it uses genes to differentiate cells. Now remember, each cell has a complete complement of the whole genome. All 23 homologous chromosomes. So it's not like one cell only has a few for it and They all have the same thing. And part of gene expression is how you get cells to differentiate. So in a muscle cell, right here, certain genes will turn on and off. And you'll end up with a muscle cell. These are all little, in each cell there's a nucleus. Versus a neuron, let's say, where they look different, they function differently, and each one, of course, is made from different proteins. So one of the things that gene expression does is it tells which kind of cell is being made, and especially during development, you want neurons in certain places and muscle cells in others. And that's kind of what one of the things it does. So gene expression is actually where everything is actually happening now. I guess it's late. Why don't we take a little break, and then we'll get back to this. Oh, I'm going to put up another top hat, too.